Filtration. Now, let's see, before we get there, let's talk about a few different things here. Blood supply comes into the arteriole, into the capillary, leaves by way of the venule. The capillary has a built-in pressure, which can be measured. Outside of the capillary, we have the interstitial fluid between the cells in the tissues, which can have a pressure. What we're going to look at is the two opposing pressures, how they work with one another or work against one another. So we're going to call the pressure inside the capillary tube the capillary hydrostatic pressure. The pressure outside of that in the interstitial space we're going to call osmotic pressure. Osmotic pressure. Now, the capillary fluid or pressure inside the capillary can be greater than that of the osmotic pressure, which means if that's the case, things can move outside of the capillary, diffuse into the interstitial space. Whereas the opposite is true if the interstitial fluid has got a greater pressure than what the capillary hydrostatic pressure is. If the osmotic pressure is greater than the hydrostatic pressure, then things can move inside the capillary from the interstitial space. And that's what we're going to be talking about as I go back. Some more understanding here. If the capillary pressure versus the osmotic pressure is the same at 25 millimeters of mercury for hydrostatic pressure and 25 millimeters of mercury of osmotic pressure, nothing's going to move either way as far as pressure gradients. However, if the hydrostatic pressure is 35 millimeters of mercury versus the osmotic pressure being 25 millimeters of mercury, we've got a difference of 10 on the side of the hydrostatic pressure. Therefore, things are going to move out. We've got a greater pressure by 10 millimeters of mercury to move out of the capillary by way of hydrostatic pressure. Whereas if we come over here, we see that an example, if our hydrostatic pressure is 18 millimeters of mercury and our osmotic pressure is 25 millimeters of mercury, we've got a difference of -70 or -7, meaning that our hydrostatic pressure or our osmotic pressure is greater than our hydrostatic pressure. Therefore, things are going to move back into the capillary from the interstitial fluid. We call things moving out of the capillary based on a greater hydrostatic pressure filtration. Filtration. We call things, substances moving, or the process of things moving from the interstitial osmotic pressure side into the capillary, reabsorption. Reabsorption. So moving out of the capillary, we're saying filtration. Moving into the capillary by way of a greater osmotic pressure than hydrostatic pressure, we're going to call that reabsorption. Reabsorption. Now, keep in mind, as we filtrate things and reabsorb stuff, we actually filter more than we reabsorb. So what's happening here? Where'd the rest of it go? If we're filtering 24 liters per day and we're reabsorbing 20.4 liters per day, we're missing something. Where'd it go? Well, lymphatic vessels flow and track along with the arteries, arterioles, as well as the veins and the venules. Lymphatic vessels. And when this fluid comes out here into the tissues, some of that fluid is picked up by the lymphatic vessels, which are in that capillary bed as well. So 3.6 liters per day flows into the lymphatic vessels, which then filters it through the lymph nodes, and then takes it back and puts it into the venous system around the neck of the body. So that's what's happening with the difference between filtration and reabsorption. Now let's go back here. So hopefully you have a little understanding what it is we're talking about. All right, filtration, driven by hydrostatic pressure. Water in small solutes forced through the capillary wall leaves larger solutes in the bloodstream. Reabsorption, the result of osmosis. Higher solute concentration leads to higher osmotic pressure of the solution. Blood colloid osmotic pressure, pressure required to prevent osmosis, caused by suspended blood proteins that are too large to cross capillary walls. This picture is simply showing us the capillary hydrostatic pressure pushing small ions and solutes through the openings. That's pretty much what we see here. Interplay between filtration and reabsorption ensures that plasma and interstitial fluid are in constant communication and mutual exchange. Accelerates distribution of the nutrients, hormones, and dissolved gases throughout tissues. Assists in the transport of insoluble lipids and tissue proteins that cannot cross the capillary walls. Carries bacterial toxins and other chemical stimuli to lymphatic tissues and organs. Filtration and reabsorption are affected by net capillary hydrostatic pressure, which is in the capillary, and net capillary colloid osmotic pressure, also in the capillary. Factors that contribute to net capillary hydrostatic pressure. Capillary hydrostatic pressure, interstitial fluid hydrostatic pressure. Tends to push water and solutes out of the capillaries into the interstitial fluid. Net capillary colloid osmotic pressure is the difference between blood colloid osmotic pressure and interstitial fluid colloid osmotic pressure. Pulls water and solutes into the capillary from the interstitial fluid. The NFP, the net filtration pressure, the difference between the net hydrostatic pressure and the net osmotic pressure. Capillary exchange. At the arterial end of the capillary, fluid moves from the capillary to the interstitial fluid. At the venous end of the capillary, fluid moves into the capillary from the interstitial fluid. And we called that filtration and reabsorption. Fluid moves from the capillary to the interstitial fluid. Filtration. Fluid moves into the capillary from the interstitial fluid. Reabsorption. Transition point between filtration and reabsorption is closer to the venous end than the arterial end. Capillaries filter more than they absorb, which we've already discussed, because they get picked up. Some of that product gets picked up by the lymphatic vessels. Capillary dynamics. Hemorrhaging reduces capillary hydrostatic pressure and the net filtration pressure. Hemorrhaging reduces capillary hydrostatic pressure and the net filtration pressure. It increases reabsorption in the interstitial fluid. Dehydration increases blood colloid osmotic pressure, which accelerates reabsorption. If capillary hydrostatic pressure rises or blood colloid osmotic pressure declines, fluid moves out of the blood builds up in the peripheral tissues, and we call that edema. Tissue perfusion. In talking about tissue perfusion, you may have heard me say that if mom and dad and grandma and grandpa are sitting in the recliners watching TV all day and never do anything and never move, that's not good for the body, because we're not driving enough oxygen deep into the tissues to satisfy the needs of the organs and tissues. The body wasn't meant to sit and do nothing. We have to use that diaphragm to pump, to actually pressurize the system, to move that chemistry in fluid form deep into the tissues so we can utilize the oxygen. And that's what we're talking about here, is tissue perfusion. The blood flow through the tissues carries oxygen and nutrients to tissues and organs, carries CO2 and wastes away, affected by cardiac output, peripheral resistance, and blood pressure. When certain cells become active, circulation to that region must increase. Cardiovascular regulation ensures that blood flow changes occur at an appropriate time in the right area and without changing blood pressure and blood flow to vital organs. Vasomotion, the contraction and relaxation cycle of the precapillary sphincters. Vasomotion. Again, when we say vaso, think in terms of vessel. Vessel. Causes blood flow in the capillary beds to constantly change routes. Controlling cardiac output and blood pressure, we have three that we can look at: autoregulation, neural mechanisms, and endocrine mechanisms or chemical mechanisms. Autoregulation causes immediate, short-term localized homeostatic adjustments. Neural mechanisms respond quickly to changes at specific sites, and endocrine mechanisms direct long-term changes. So, controlling cardiac output and blood pressure through means of either autoregulation, neural mechanisms, endocrine mechanisms, or a combination thereof. Looking at autoregulation and homeostasis, if we have a stimulus which disrupts homeostasis, producing an inadequate local blood flow and blood pressure, the sensors pick up the information, send the information to the brain sends out the effectors. The effector here is going to be the precapillary sphincters, which respond, local decrease in resistance, and increase in blood flow, which in fact takes us back to homeostasis by increasing blood flow. So our effectors are going to be the precapillary sphincters, short-term, local, are going to open up a bit and let those capillaries have a little bit more blood flow. If autoregulation does not restore homeostasis, we're going to go to either the neural or the endocrine system or combination. Here we're looking at the neural mechanisms to get back to homeostasis. The effector is the brain. Stimulation of the receptors sensitive to changes in systemic blood pressure or chemistry. Activation of the cardiovascular center. The cardiovascular center in the medulla oblongata. So the brain is basically going to talk to the activation center in the cardiovascular center of the medulla oblongata, creating a short-term increase in blood pressure by sympathetic stimulation of the heart and peripheral vasoconstriction, taking us back, increasing blood volume and blood pressure, taking us back to homeostasis. Whereas if we take the endocrine route by way of the kidney, We're looking for a long-term increase of blood volume and blood pressure, getting us back to a blood increasing the blood volume and pressure. The kidney will be releasing hormones necessary to accomplish that. Autoregulation of blood flow within tissues adjusted by the peripheral resistance while cardiac output stays the same. Autoregulation adjusted by peripheral resistance while cardiac output stays the same. We're basically closing those or opening those sphincters or closing them. Cardiac output stays the same. Precapillary sphincters are stimulated to constrict or dilate, altering blood flow. The local vasoconstrictors reduce blood flow by constricting precapillary sphincters. Okay. Autoregulation of blood flow with tissues. Vasodilators are factors that promote dilation of precapillary sphincters, increasing blood flow. Here's a list of some of those local vasodilators. Neural mechanisms. The cardiovascular center of the medulla oblongata consists of cardiac centers and vasomotor center. The cardiac centers are the cardioacceleratory center, increases cardiac output. The cardioinhibitory center reduces cardiac output. And then the vasomotor center controls vasoconstriction, controlled by the adrenergic nerves, stimulate contraction of the arterial walls, control of the vasodilation, controlled by the cholinergic nerves, relax smooth muscle. Then we have vasomotor tone, produced by constant action of the sympathetic vasoconstrictor nerves. Reflex control of the cardiovascular function. Cardiovascular center monitors arterial blood. We have baroreceptor reflexes and chemoreceptor reflexes. Baro meaning pressure. So we have receptors sending sensory information based on pressure, and chemoreceptors sending information based on the chemistry that's flowing through the area. Baroreceptors respond to changes in the blood pressure, while the chemoreceptors respond to changes in chemical composition, particularly pH levels and dissolved gases. The baroreceptors, stretch receptors in walls of the carotid sinuses, maintain blood flow to the brain, and the aortic sinuses monitor start of the systemic circuit. Right atrium monitors the end of the systemic circuit. So we have baroreceptors, pressure receptors, sending information strategically placed. These receptors are strategically placed in areas such as the common carotid artery and placed in the bifurcation where it turns into a Y, branches into two different arteries. It'll be placed right there in the center, so it monitors the pressure that's hitting that point. So changes in blood pressure in the ascending aorta trigger the aortic reflex. It adjusts blood pressure and flow in the systemic circuit. When blood pressure rises, cardiovascular centers decrease cardiac output, cause peripheral vasodilation. When the blood pressure falls, cardiovascular centers increase cardiac output and cause the peripheral vasoconstriction. Keep in mind when we're talking about this that we indicated that there's more volume of blood in the venous supply than there is in the arterial supply. Okay, the arterial baroreceptors monitor blood pressure at the end of the systemic circuit. The Bainbridge reflex responds to stretching of the wall in the right atrium. So what this picture is showing us is our normal range of blood pressure. Stimulus has taken us away from homeostasis by increasing the blood pressure. We don't want the blood pressure increased. Therefore, we're looking at the baroreceptor reflex of the carotid and aortic sinuses. So it's saying the baroreceptors of the aortic and carotid sinuses are stimulated. That information, it's a reflex, it's going to go to the effector, the medulla oblongata, which houses the cardiovascular center, both cardioinhibitory and cardioacceleratory centers. And so what we look here, as well as the vasomotor center is inhibited, resulting in, as far as the cardiovascular centers, decreased cardiac output to the heart, and vasocenter is inhibited, creating vasodilation of vessels, reduced blood pressure, both leading to reduced blood pressure, restoring homeostasis, and decreasing the blood pressure. Now we can go that way. If blood pressure is increasing against what homeostasis was, or we can go say that homeostasis has been disrupted and blood pressure is decreasing. We see the route, it's basically the same route, doing the opposite to get back to homeostasis. Chemoreceptor reflexes. Chemoreceptor reflexes, as opposed to the baroreceptor reflexes we just went through. Chemoreceptor reflexes. Peripheral chemoreceptors in the carotid bodies and the aortic bodies monitor the blood. Central chemoreceptors below the medulla oblongata monitor cerebrospinal fluid, control respiratory function, and control blood flow to the brain. Respond to changes in pH, O2, and CO2. Coordinate cardiovascular and respiratory activities. So in the chemoreceptor reflex, we have increasing CO2 level and decreasing pH and O2 levels. Not good. We want to affect the chemoreceptors. We want to stimulate them. Medulla oblongata, respiratory centers are stimulated, resulting in increased cardiac output and blood pressure. With the respiratories stimulating, results in the respiratory rate increases. With the vasomotor center stimulated, it increases or creates the vasoconstriction, increasing the cardiac output and blood pressure, leading to increased pH and O2 levels and decreased CO2 levels. Decreasing and increasing. Okay, so we're getting back to homeostasis. Cardiovascular centers and central nervous system activities. Through processes and emotional states, we can elevate blood pressure by cardiac stimulation and vasoconstriction. Endocrine mechanisms. Hormones have short-term and long-term effects on cardiovascular regulation. For example, epinephrine and norepinephrine from the adrenal medulla stimulate cardiac output and peripheral vasoconstriction. Antidiuretic hormone released by the neurohypophysis or the posterior lobe of the pituitary elevates blood pressure, reduces water loss at the kidneys. ADH responds to low blood volume, high plasma osmotic concentration, and circulating angiotensin II. Let's just discuss this a little bit. Antidiuretic hormone. Diuresis means we're losing fluids from the body. If we want to anti that, we're not losing fluids from the body. So we're retaining fluids. If we're retaining fluids, That reduces the water loss, elevates the blood pressure as we're holding those fluids in. So ADH responds to low blood pressure because we want to increase it, therefore holding the pressure or the fluids back in the body. And while I'm saying that, remember, water will go where sodium goes. So if we're retaining sodium, we're retaining water. Okay, high plasma osmotic concentration, circulating angiotensin II. Angiotensin II releases in response to a decrease in renal blood pressure. Renal blood pressure. Stimulates aldosterone production, antidiuretic hormone secretion, thirst, cardiac output, and peripheral vasoconstriction. We've talked about aldosterone. Aldosterone and ADH are pretty much doing the same thing. They're both designed in their own way to help retain the fluids of the body. Erythropoietin, released by the kidneys, responds to low blood pressure or low oxygen content in the blood, stimulates vasoconstriction and red blood cell production. So basically what we're doing with erythropoietin is we're increasing the volume of the blood. Released by the kidneys, responds to low blood pressure or low oxygen content in the blood. We need more oxygen. We need more blood pressure. Let's make some more. Stimulates vasoconstriction and red blood cell production. Erythropoietin, the hormone, erythropoietin. Okay, the hormonal regulation of blood pressure and blood volume. Well, let's see, we're going to start here. We can either increase blood pressure and volume or decrease blood pressure and volume as far as the stimulus is going to affect either one of those. Okay, I'm going to go through the process, showing you baroreceptors versus kidney, sympathetic, heart and blood valves versus the hormone release. And it's basically what this chart is saying is what we've already been through. So however you learn best, you can either use the chart or take a look at what we've gone over here or use your own notes. Natriuretic peptides. We've discussed them in endocrine. The natriuretic peptides, we have two of them. The atrioneuritic peptide, the ANP, produced by cells in the right atrium, and the brain-neuritic peptide, the BNP, produced by the ventricular muscle cells. Respond to excessive diastolic stretching. Reduce blood volume and blood pressure by several means. So here we see the hormonal regulation of blood pressure and blood volume by way of BNP and ANP. Let's see, let's just kind of follow that again. Either way. Increased sodium loss in urine, increased water loss in urine, produce thirst. Inhibition of ADH, aldosterone, epinephrine, norepinephrine release. Peripheral vasodilation, decreased blood pressure, and blood volume. So all the things that we talked about as far as increasing blood pressure, it's basically creating a situation where it's preventing that. Okay. Again, you can follow these or use the information that we've gone over. Blood, heart, and cardiovascular center work together as a unit, respond to physical and psychological changes, exercise, blood loss, maintain homeostasis. Vascular supply to specific regions. Some organs have separate mechanisms to control blood flow. 3 important examples are the brain, the heart, and the lungs.